Hello, everyone. Thank you, everyone, for joining this installment of Boyce Thompson Institute's Breaking Ground discussion series. My name is AJ Bushy, and I'm in the communications department here at BTI. And for this month's Breaking Ground, we welcome BTI adjunct faculty member Miguel Pineros. Hey, Miguel.
Hey, AJ. Thanks for having me.
Yeah, thank you so much for joining us. And Miguel's a plant physiologist at the U.S. Department of Agriculture, right across the street at the Holly Center up here on Tower Road. And he's going to talk with us today about using electrophysiological techniques to study how plants adapt to changes in the environment. So let's kick things off. Miguel, can you tell us a little bit about yourself and how you came to be here in Ithaca now?
Since 1996. Yeah, thank you, AJ. Thanks for the invite again. I was born in South America. I was born in Bogota, Colombia. So I grew up there, big city. I was thinking the other night, I'm not sure where my infatuation for plant biology came from, but I was thinking it probably came back somewhere in the middle of high school. I used to hijack my brother who used to study medicine. I used to hijack his biochem lab practices. And it consisted of doing experiments with frog legs and seeing how you could actually excite the frog leg to make it sort of contract. So I think that's probably where I got my thing for biology. So I went to the University of Los Andes in Bogota. I studied biology there. I did a BSA. Then I did 1 year as a high school teacher. I studied physics. I also taught math, biology, and then I dropped that and I went to work 1 year for the Smithsonian as a research assistant doing tropical ecology. And then in '92, I took a big turn and decided to do a PhD. I took an offer from the University of Adelaide in Australia, and I went there to do my PhD with Mark Tester. And it was a 180-degree turn because I moved from ecophysiology to doing basically studying biophysics of plant membranes transporting plants. So that was my first training with biophysics. And finishing my PhD, I was contacted by Liam Koshin here at the USDA, and he was interested in hiring somebody for a postdoc, trying to implement those techniques to understand how plants actually adapt to the environment. So he wanted to integrate those sort of techniques with more sort of ag-related questions. So that's how I ended up coming in '96 to do a postdoc at Cornell. And ever since I've been here, I worked for Cornell, then I moved to USDA, and through the years I became a BTI adjunct as well. So that's pretty much how I ended up landing from Colombia all the way to Ithaca.
Yeah, and you mentioned, electrophysiology and cell membrane transporters. Can you tell us a little bit more about that? Like, what are these transporter proteins and what do they do in plants?
Sure. So what I brought was that sort of knowledge of biophysics, right? And can we go back one slide, Mike, please? Thank you. So I brought that knowledge of sort of biophysics and tried to integrate it with plant physiology, biochemistry, cell physiology. In terms of biophysics and studying membrane transport, we were interested in understanding how these transport proteins, which are located at the membrane of the cell, are involved or mediate responses in regards to how do plants deal when there's too much of something, a toxicity, or how do plants deal with there's not enough of something, a deficiency. So this, you can imagine a cell membrane, right? It's sort of the analogy normally is, you talk about like a fortress, right? The fortress is surrounded by this wall, which is your plasma membrane, it's your lipid, it's not permeable to things. And the transporters could be the gatekeepers. They're the ones that actually allow, ions or nutrients or signals to move in and out of the cells. So there are different ways of studying these transporters. And what we've been doing through the years, or one of the approaches we've been using is precisely electrophysiology approaches. And if you want to move one slide, Mike, that'll be great. So basically, what is electrophysiology? What you're doing is you're just studying the electrical properties of biological cells and tissues. And it involves measuring either voltages or electric currents that occur at different scales, right? It can occur at the whole organ scale. You can look at them at the single cell scale or at the single protein. Probably what you're most familiar with would be what I'm showing you on the, the left up panel, which is your regular EKG when you go to the cardiologist, right? They put a whole series of electrodes in one arm, in your leg, in your chest, and they're measuring the electrical activity that the heart produces. And they're measuring it at the whole organ level. Just like I show you at in the right panel, right? In plants, you can do exactly the same. You can put these surface electrodes, right? And measure the activity or the electrical activity that occurs across an organ. There are some organs that have fast responses. For example, the mimosa pudica or the fly trap. They're very fast events that occur, but you can also measure changes in those electrical properties at a lower scale. It's what we call surface potentials, and it's how do those electrical potentials change as the plant is responding to a change in the environment. That's sort of your whole organ level. Then in the 60s, there was 1940, 1950, there was a big break by two British investigators, Huxley and Hodgkins, and they were able to work with this big cell. It was a squid axon, and they were able to put in a very fine glass microelectrode. And that was the first time that we were able to record what happens electrically, not in a whole tissue, but in a single cell.
And how big are these cells? It sounds like they're very big.
The axon cells are pretty big. They're about 1 millimeter in diameter, which, you know, you can see it by the eye. Considering that's a single cell, that's pretty big and it's electrically very active.
Yeah.
At the same time, people were able to do it in plant cells too, right? What they use these giant algal cells, Caracorellina, right? There's also big single cells from node to node, and you're able to put these glass electrodes in and measure the electrical activity or the changes in potential that happen due to transport activity in a single cell. Through the years, we've become more and more sophisticated, and now you're able to create very fine glass microelectrodes and go and study single cell responses in isolated cells from other plant cells, right? And you can go even more than just looking at single cell activity. You can look at single proteins, and if you want to move Mike one slide, right? And this is what fascinated me. I mean, that's really why I got hooked into this. And you can look at the activity of a single molecule. And let me just take you through very quickly. What you see in the cartoon on your left is a little device. It's a two-chamber system, which is divided by a very small hole. It's about 100 microns in diameter. Across this hole, you can take an artificial lipid, which is deprived of any protein, and you can create an artificial lipid bilayer, right?
Like a membrane.
Then what you can do is you can take a membrane, right? What you can do is you can take a protein of interest or a transport of interest and incorporate it or reconstitute it back into that bilayer. And what you can actually look at, or you can actually record, is the transport of the ions that take place across the bilayer through the transporter, right, in real time. And what I'm showing you at the right side is an example of what the output looks like. And what you see is an oscillating current in very discrete steps. What you're seeing there is a single molecule. In this case, there's actually two molecules in that little patch of membrane, but they're opening and closing in a very discrete manner. So real time, you're looking at the activity of a single molecule. And you can actually change its properties of how fast it opens, how fast it closes. You can look at what pharmacological agents can be applied to change its activity. And that's the kind of resolution we're able to obtain electrophysiologically nowadays. And this has been going on since middle of the 80s, probably. But to me, that was the most fascinating part of the electrophysiology, which was you can look at a single molecule working real time right there next to you in the bench.
Yeah. So what I noticed the EKG looking thing, not sure what it's called. It says close, opened, and two opened. What is two opened? Does that mean there's two proteins that are opening?
Right, the two, in the example I'm showing you there are two proteins sitting in that little patch of membrane.
Okay.
Right. So what you see, one open is when just one of the two proteins are open. So you see a much larger current or a much larger flow of ions. And when you see two steps going down, is the two proteins open at the same time.
Gotcha. Okay, so you're actually measuring the ions that are flowing from one tank to the other.
That's correct. You're measuring the movement of ions from one of the chambers to the other side of the chamber.
That's really cool. And so how has the study of plant membrane transporter proteins evolved over the years?
Well, it's taken a big leap since I started doing this. I mean, you can think earlier, before the 90s, right? I mean, the physiologists, knew a lot about how plants respond to certain changes in the environment, right? However, we had characterized the physiological mechanisms, right? But in many cases, we didn't understand the molecular nature of what mediated these responses, right? So in terms of transport, for example, we started looking since I would say probably the mid 80s, right? On how do the plant cells respond to certain environmental cues. And we use all these electrophysiological techniques, right? So we could isolate, say, a protoplast and put an electrode and look what happens when it's exposed to a high salt environment and see what kind of membrane transporters would activate. But this was all a physiological or electrophysiological characterization. I think the big breakthrough comes when we get all the sequencing data, right? And now we're able to associate genes, right, which underlie this particular membrane transporters that we were studying. And now we can associate a particular functional characterization that we've done of an unknown transporter. We can tag it, we can point it to a particular gene. And this was the big breakthrough, I would say, late 90s, early 2000s. What that has allowed us to do now is start to characterize a whole family of transporters, right? And we can take these genes, we can express them outside the plant. We can use heterologous systems. We can put them into, say, mammalian cells or frog cells or something, but we can look at their activity of how they work, right, by expressing these genes, and we can characterize how this gene or this particular family of transporter works. Then came the breakthrough of structural biology, I think, and it's now we're starting to get more and more knowledge, not just about the function of these genes, but about the structure of the proteins that are encoded by these genes. Now, it's been a big task, right? You can consider, you can think that membrane proteins in general make about 40% of the total proteins of the proteome in eukaryotic and prokaryotic cells.
Oh, wow.
Right?
I didn't know that.
So, however, right, there's only 2%, more or less, of the structures that we have out there, right, nowadays, are of membrane transporters or membrane proteins, right? So we're leaping behind a little bit. You can imagine the first structure, the first crystal structure, for example, of a soluble protein was determined in 1960, and it was for the globins, for the hemo and the myoglobins. And it was not until like 25 years later that we were able to get a structure, a crystal structure, a high resolution structure of an integral membrane protein. And this was actually the photosynthetic reaction, right, of a bacteria. So membrane biology, membrane proteins are very difficult to work in terms of generating high resolution structures. However, with the appearance of cryo-EM, we're making a lot of progress nowadays in terms of the amount of new structural information we're getting on many of these membrane transporters.
Yeah.
So now we have the structure, we have the function, and now we can start playing and seeing what are those structural functional relations, right, in this particular transporters.
Yeah, I was gonna say, I seem to remember it being difficult to get to crystallize and get the structures of membrane bound proteins in their natural state. Because if you purify them first and then crystallize them, that's not their actual structure when they're in the membrane, right? So is that, you mentioned cryo-EM, what is that?
Well, that's relatively recent. Is a breakthrough. Yes, in the old days, what you would do would be you would crystallize. You would obtain a crystal of your protein,
Mhm.
if you were lucky, and you would do X-ray crystallography to basically determine how that structure is composed. Nowadays, with cryo-EM, what they do in a rough sense is you purified your protein, you reconstitute it, right? And you're basically just taking images and you're averaging a lot of images of what your microscope is allowing you to see, right?
Okay.
And with that, you reconstruct a 3D image or a 3D reconstruction of what you're getting, and you predict the structure
I see.
in a nutshell.
So you don't have to crystallize.
You don't have to crystallize it, right? Just
Very cool.
it's
And
a
so
big
you
breakthrough.
mentioned, with the discovery of genes and molecular biology, how is that changing your work and what you do? And it seems like
Well,
that's a big difference.
yes, I think I've evolved a little bit with just like the field has evolved,
Yeah.
right? And I'll just give you an example of like our most current and long-term research that we've been doing. And Mike, can I get the next slide, please? Thank you. So when I got here, I was involved in a project to work in aluminum tolerance, right? And let me take you back a little bit to physiology. Aluminum toxicity is a big problem throughout the world. Why? Because aluminum is bound in the soil to all the silicas, right? It's not a big problem. However, when the soil goes to low pHs below 5, what happens is that aluminum just falls out and becomes soluble in the rhizosphere, right? And that soluble form of aluminum, Al3+, is very toxic to the plants. So you can see a picture there in the middle, for example, there's a little wheat plant, right? Which, when exposed to aluminum, the roots are just totally inhibited. Aluminum inhibits root growth, it inhibits water uptake, mineral nutrition. But there's some variability in terms of the cultivars. There's some cultivars that are resistant. There are some cultivars that are sensitive. That we knew earlier in the 90s, right? Then in the mid-90s, what we found out was that those varieties which are sensitive, right, is actually due to a lack or a reduced amount of exudation of organic acids. So the really tolerant varieties, what they're doing is once they're exposed to aluminum, they release all these organic acids. What do the organic acids do? They bind the aluminum, they chelate it, and they form complexes that are not toxic to a plant. So that was great. And again, we had characterized the physiological mechanism, right? Now, what is the nature of the mechanism underlying that process? That's what I sort of came in late 90s, and we started looking at single cells from roots. We use something called a patch clamp technique, which I show you in that sort of a middle panel. With one single electrode, we can study the activity of a single cell that we've isolated. We remove the cell wall, and we're looking at the electrical activity of a protoplast. And what we were able to identify was that those little traces that you see next to them is in the absence of aluminum, minus Al, right? There's not a lot of currents. There's not a lot of activity of transport. When you put aluminum in the solution, you get a lot of currents. You get a lot of transport. So now in the cell, we were able to identify a mechanism that can account for that physiological observation that we saw at the whole plant level, right? However, we don't know which protein is carrying that current or that transport of ions. Then came in the genetics, right? Then it came all the sequencing, and we bumped into the genes, right? Which are two predominant families, the LMT families and the MATE families. They stand for different things. They're very different families, but they achieve very similar functions. So now we have a hold of the genes that actually encode those functional properties that we're observing. Now we can take those genes and we can express them in different systems. I'm showing you at the bottom panel, we use this frog eggs, this Xenopus eggs, where we can actually express the protein and come with electrodes and study the activity of the protein. So now we could study the protein that's encoded by these genes. We could study their functional properties, how they behave, what do they transport, what antagonizes with them, right? And the nice thing is we have the DNA, right? So now we can start predicting, maybe this part of the DNA encodes this particular part of the structure. And we can start doing a lot of functional structural analysis, which is we can alter the DNA and see what the change in the functionality is. And we can start making predictions of what the changes in the structure might be. And nowadays, a couple of years ago, finally, there are two groups that came out with cryo-EM structures for that ALMT family. So now we can take all our functional data and all those mutations we did and all those permutations we did and look what is it really in the structure of the protein that we had been affecting. So that's just an example of how the field moved, or I moved along with the field, I would say.
That's really cool. Are you working on any other projects similar to the loom tolerance work?
We have a couple of other projects going on related to this ALMTs. There's some nice work by Liang Cheng and Senom Zhu here at Horticulture in Cornell. They approached me about, I would say, a good 5-8 years ago. And they say, Miguel, what do you know about ALMTs? And I said, well, I don't know. I work with aluminum tolerance related ALMTs. And they said, well, we work with Apple. And there's a single gene, right, that might explain the difference between sweet apples and very acid apples. A single gene, they looked at it from the genetics point of view, they identified a QTL and they said, well, it looks at this particular gene can be the determinant, right? What turned out to be is a transporter. It's an ALMT, and it's localized to the tonoplast, to the vacuole, right, of the cell. So what it does is it mediates the movement of organic acids, of malate, into the vacuole, right? And there is one allele, one type of transporter that determines acidity, and there's one type of transporter that if you have that type of transporter, then you're acid. So what we did is, can you go back one slide, Mike, please? So what we did, that gene is called MA. So what we did is we actually look at the two genes. We said, okay, we'll take the gene and express and characterize each gene separately and see why are they different in their transport. And that's what we've been doing. We're looking at is structurally, why are they different, which will explain functionally why they're different, and which would explain why one is acid and why is sweet. And we're looking at its regulation and how it's worked. So it's work in progress since.
Yeah, that's interesting. So if it do you know if it pumps was it malic acid? Is that what it would be?
Yes.
If it pumps malic acid into the vacuole, does that make it acidic or sweet?
It makes it acidic.
Makes it acidic.
Right.
Okay. I guess that makes sense. Malic acid would make it acidic.
And the other gene is the big difference with the other gene is the other gene encodes the same transporter, but it's truncated. It's missing a little bit, right, of the protein. So apparently the transporter is not as functional or is not functional.
So it doesn't pump as much, probably.
Exactly.
Okay. Oh, very cool. So the research you're doing, ultimately, how will it benefit agriculture?
So I think that the type of research we're doing is providing nowadays providing a structural functional platform, right? And by that, what do I mean? It's allowing us to identify, right, identify transporters that mediate a particular phenotype or a particular trait that you're interested in, right? and study how that gene works. But it's also allowing us to, like with the example that I gave you with the apple, it's allowing us to study the difference in alleles, right? So basically at the protein level, we're being able to characterize the natural variation that occurs in a population, right?
Mhm.
And ultimately, I think what this would lead is like, with all these advancements in being able to, edit the gene relatively easy. What I believe the future is, we should be able to develop strategies to actually engineer the transport. If you know that this particular amino acid and this amino acid cause this change in the protein, why not go in and just change that particular acid and that particular amino acid at the genome level, right? And this should give us a handle, right, to be able to generate phenotypes or generate plants where we enhance those traits that we desire. And in particular case of what we're studying is, we want plants that should be able to tolerate harsh environments like acid soils. We want to improve plants. We want to improve their tolerance so that we can start using those marginal regions to increase productivity.
Right, so like those loam tolerant corn plants, could you make them even more tolerant or something theoretically?
Yes, you can, right? And it has been shown that if you take this particular gene and you overexpress it, you produce a lot of this protein in very sensitive cultivars like barley, you enhance the tolerance and they're able to grow in this particular nasty conditions.
Oh, that's cool. Okay, if folks have any questions, feel free to put them in the chat box there, and we can start the Q&A portion. While we're waiting, I do have some other questions. How is climate change impacting your work? Is that at all even?
I think in every sense. I mean, I've been involved in abiotic stress for the most part of my research. So we need to start generating some information in regards to how can we exploit this particular genes, this particular transporters, to enhance the properties of or the tolerance of plants to this ever-changing conditions, right? Yeah. Heat stress, drought stress, any climate change, is going to require a change in the plant, right, to be able to tolerate this condition. So in every sense, every change in abiotic stress that we obtain is a new target. It's a new field where I think eventually we'll have to move into.
Yeah. Is there any potential of this to work on like disease resistance or like insect resistant, or is it really more like abiotic, as you said, abiotic?
No, it's totally. We have ongoing projects. There's a student here, Nathan. He's working. It's another collaboration we have at Cornell. There's a bug, right, that basically when it infects the plant, right, what the bug does is it upregulates the expression of a plant transporter, right? It's a sulfate transporter. But so why is it that this bacteria is really targeting the upregulation of this transporter? Obviously, it's asking something from the plant. It needs something from the plant that this transporter is giving it, right? So we're in that sort of trying to understand what is it that this transporter does in the plant, and what is it that it gives the bacteria, right? There's other, all those pathological responses ultimately involve transporters, right? Because transporters not only carry ions in, ions out, and metabolites, but they carry a signal. They are involved in a lot of the signaling cascades in plants. So they're used to sort of transmit the information. So you can imagine an infection, a pathological process happening here, right? Well, all these transporters are the ones that basically transmit that message throughout the plant.
Yeah, that makes sense. Okay, so we've got a few questions in the chat box. One from Lizzie. Hi, Lizzie. Can the methods you use to characterize the function and activity of plant transporters be used in yeast to characterize fungal transporters? That's a good question.
Absolutely, yes. All the electrophysiology basically looks at cell physiology, call it plant, yeast, bacteria. Yeast, you can do single cell analysis of transporters. Obviously, if you know the yeast genes, right, you can express those genes in other systems and study the activity of those proteins in other systems. But it's totally amendable to any system.
That makes sense. Here's one from Java Harry. I hope I pronounced your name correctly. Cool science. How selective are the transporter proteins? Do we know the transporters in Brassicaceae? Do you look at electric charges and acidity, but how neutral molecules are being studied?
Okay, couple of questions, right?
questions.
How selective are they? That's a big field at the moment, right? Because once we have all this genome, right, we had so many transporters to look at, and we said, okay, let's look at the sequences. And we look at this sequence and we go like, okay, they group together, they're a family. What do they do? We don't know. Let's compare them to the animal systems, right? And we go and we look there and it says, oh, sequences that are similar to this in animal cells, they work like chloride transporters. So the plant people said, oh, these are chloride transporters. And we're starting to find is that not necessarily, right? Not sequence identity doesn't mean functional identity. So that's something we look at, and it's very complicated to establish how selective they are, right? Because in many cases, if you take a transporter and you feed, I would say, but if you expose it to, say, cadmium, it transports cadmium. So I say this is a cadmium transporter, but somebody else comes down the road and puts nickel, and it transports nickel, and they say this is a nickel transporter, right? So we're in that age in science where we really have to tackle that question more carefully, which is fine, it can transport the two, but in the plant system, under field conditions, what will the transporter do? And that's more difficult to study.
That's fascinating. Yeah, I remember there's I read a paper recently that kind of touched on that, but I don't really remember anything beyond that. So, so what about the next one? Do we know the transporters in the Brassicaceae plants?
I have to say, I'm not aware of a lot of characterization done on them, right? We tend to use very sort of either crops, obviously for the agricultural importance, or model plants. There's also obviously a lot of research going on like Arabidopsis transporters, and so I'm not aware, I have to say, of how much research there has been in Brassicaceae.
And you look at electric charges and acidity, but are neutral molecules being studied?
Yes. So the example, most of the example that I gave you, right, was sort of ion channels, right, which are transporters which move ions, right? There are other types of transporters, right? They are transporters that move non-charged compounds or molecules, right? They're transporters that not just behave like a channel that, the ions flow through, right? Some transporters require energy to move those substrates against a gradient, against an electrical gradient, against a chemical gradient. Those are all those pumps, right? Which use ATP and they pump those compounds. So yes, they don't have to be ions. They can be neutral molecules. They can be complex secondary metabolites, right? So for the electrophysiological approach to work, right, there has to be a movement, a net movement of charge. That's why most of the transporters that we study are ion channels, right? Because they transport charge. But they're transporters that are neutral molecules, sure.
So how would you study those from an electrophysiological standpoint if there's no change in voltage or?
It depends. If it's totally neutral, you can't, right? There are other transporters, for example, sucrose transporters, right? They work in such a way that it's not just sucrose moving, but it's coupled to something. So what the transporter does is it uses the proton gradient, proton movement, right, to couple the movement of sugar with it, right? So electrophysiologically, what you can look at is the movement of protons when you have sucrose or when you don't have sucrose, or right. So just so long it's coupled to a charge movement, you can do it.
Could you also look at like ATP turning into ADP or something?
Absolutely. Like that? All the traditional biochemical ways you can do it.
Uh-huh.
Electrophysiology is just a different approach to techniques to look at these problems, right? You can use reverse genetics, forward genetics, look at phenotypes, look at changes in concentration of ions in your tissue. You know, by no means this is the only way of looking at membrane transport. It is the way of looking at more at the cellular level.
Right. All right, very cool. Here's one from David Silberman. Hi, David. Can this electrophysiology approach help identify the sequences responsible for the different flavors that distinguish the unique tastes found among apples and other fruits? That's a good question.
Yes, sure. And what we do is we say, well, this particular genes, right, determine the flavors in apple, right? What we do is we look at homolog genes, homolog sequences, the orthologs of them, right, in other species, you know, in pear, and basically look at those genes, and we can actually take those genes and perform the electrophysiological analysis of those genes and see if they behave in the same way. For example, this apple ALMT9, right? Nice. It determines sugar levels of sweetness in apple, right? So now what are we looking at? We're looking at the homolog, but in grapes, right? We want to see, is there a correlation of the different versions of ALMT9 in grapes that can be correlated with the different flavor, with the different sweetness, with the different acidity and flavor in grapes? And you can imagine that has a big impact in all the wine production and disease and resistance.
This one from Debbie, CFO, CF1, ATP transport, classic pH gradient experiments, Andre Jagendorf work. Study the phosphorylation. I think that's similar to what I don't think there's a question there, but that's similar to what we were talking about earlier.
Right. In relation to that, yes, we can look at, these proteins don't work all by themselves, right? They have accessory proteins, some of them, not even just ATPases, but even the channels that allow the movement of passive ions, they're regulated by phosphorylation, somebody puts a P on them and it shuts down and the protein doesn't work anymore. Somebody takes out the P and they work again. So at the protein level, yes, they're very regulated as well.
If any other folks have more questions, please put them in the chat. We have a follow-up from David. Can the sequence responsible for blueberry flavor be introduced into an apple? That's a good question. That'd be interesting.
I don't see why not, right? Sure, you could create a transgenic, bringing the blueberry into the apple one, or the apple gene into the, right? That's in the case of this ALMT transporter, that's the trait that it underlies. That's the trait that it determines. So there's no reason why bringing that gene and overexpressing it wouldn't provide that trait if it's a simple trait in apple or in berry, right? Just like we do with, for example, the tolerance, right? The tolerance to aluminum is just another trait, right? We take a gene from wheat, we put it into barley, and do we get the same trait, the same agricultural trait? Yes. So yes, I don't know to what extent you're allowed to do this kind of gene flow in fruit research, but theoretically, yes.
Yeah, there are cotton candy flavored grapes now. My daughter loves them. It's kind of weird. It doesn't taste right to me. I can't eat them. I think they're bizarre, but she loves them.
I'll stick to the Concord. Yeah.
Yeah. One more question. Is it possible to study transport between cell organelles? That's a good question.
Yes, and we've I mean a community, not me personally, but we've been doing that. Yes, in the old days, what you would do is you would actually disrupt the cell and isolate the different organelles. You could isolate intact vacuoles, for example, nice, beautiful, round intact vacuoles. You could isolate chromoplasts, intact chromoplasts. And you would do that by making a cell preparation and run a sucrose gradient. And then you would take the different fractions where the different organelles were. And you would study them. You would do electrophysiological approaches, or right. Or you could isolate those fractions and reconstitute little pieces of those fractions into artificial membranes and look for ion channel transport stuff. So yes, it's more tedious, as you can imagine, right? I mean, you're already dealing with a 20 micron cell like that. Once you open it, well, you have a very small vacuole, but beyond that, every organelle is in the micron range. So it becomes more difficult. And in that sense, I think once you take a biochemical approach, you're better off, right? Because technically it just becomes way too demanding.
I was wondering, how, I don't know if there's an easy answer to this, but how quickly can plants evolve these transporters, like to survive when environments turn harsh like this?
I think it'll take you a lot of generations.
Yeah.
Right.
Or maybe the question is, can they just repurpose an existing transporter?
I was gonna say, yeah. I mean, when you look at this ALMT, right, that mediates aluminum tolerance, right? Because we always said, oh, that's the aluminum tolerance gene, right? We discovered the aluminum tolerance gene,
right.
And it's not. It's a transporter that it's there regardless of aluminum or not, right? If you're in alkaline soils, it's there and it's working, it's doing its thing. It has a fluke, right? Which is when you expose it to aluminum, and it has to be aluminum. It cannot be any other trivalent cation. When you expose it to aluminum, the transporter becomes more active, right? And releases more organic acid than the other transporters, right? So obviously it's a fluke, but once you have the selective pressure on that fluke, right, of a transporter that was existing that performs a different function, well, it becomes, right, a transporter which acquires that particular trait or that particular tolerance, or provides that particular tolerance in acidic conditions. So right. I think just provided you have the functionality existing there, well, plants, I wouldn't say they will evolve, but they will adapt to a particular environment.
Right. So it's not necessarily an aluminum tolerance gene. It's still an organic acid transport gene, right? It's just turned up by the aluminum. So if it could be turned up by something else, then it would be whatever tolerance, right?
Right. So
very cool. I like thinking about that kind of stuff. I was an evolution guy, so then my brain always goes there. Does anyone else have any more questions? I'm not seeing any more in the chat box. Now's your chance. Let me make sure I got them all. Oh, we got one more. Oh, there's a thank you. Thanks, Miguel.
Thank you, everybody. Such
cool work. All right. Well, thank you, Miguel, for joining us today and telling us all about your really cool research. And thank you, everyone out there, for joining us out there in Zoom land. We always appreciate it. Some awesome questions today. It's always great to do these breaking grounds and have a nice, engaging audience. And if you have any follow-up questions, please feel free to reach out to me at communications@btiscience.org, and I can help get some answers. And I'm going to throw a bunch of URLs in the chat box that I'm going to be reading off. Please join us for BTI's next Breaking Ground discussion series. It will be with Dr. Michelle Heck, who is a molecular biologist with the USDA and an adjunct faculty member here at BTI. We don't have a date for that one yet, so please stay tuned. It will be sometime in late March, and when we have that date, we'll send out an email to everybody. So please keep an eye out for that one. And you can also see replays of all 30 of our previous Breaking Grounds on our website at btiscience.org/breaking-ground-discussion-series, which is in the chat box there. And you can read more about BTI Science in our annual report. You can see the URL there. And BTI is an independent nonprofit research institute, and we operate in large part thanks to the generosity of community members like you. And if you'd like to make a gift to support BTI, you can donate online at btiscience.org/give, or email our development team at development@btiscience.org. Thanks again, everyone, for joining us, and thank you huge amounts, Miguel. I really appreciate you doing this for us today. And everyone, please have a wonderful day and be well. Bye, everybody.
Thank you.